Hi guys, it's Mr. Y. Today we're going to be continuing our genetics units talking about chromosomes and DNA and how they're connected to genes. And then we're going to be learning about 3 different types of DNA mutations called point mutations, the differences between haploid and diploid, and talking about the functions and purposes of mitosis and meiosis. So some of this will be a review from the mitosis handbook, but a lot of it won't be, so make sure you're paying attention to what's familiar and what's new. All right, first off, chromosomes. Now, chromosomes exist only in eukaryotic cells. That's to say cells that have nucleuses and membrane-bound organelles. They're basically very, very compacted strands of DNA, and they only appear in cells just before the cell's about to divide, just before it's about to undergo mitosis or meiosis. So you can kind of think of them as the travel-size version of DNA. Most of the time, DNA is left in the nucleus uncoiled as chromatin. When the cell's about to divide, about to undergo mitosis, it condenses into the chromosome form. Now, it typically looks like an X structure. You can see here it has this classic X shape. And this X structure has different names for different portions. So each branch is called a chromatid. And neighboring branches are called sister chromatids. So these are sister chromatids, and then these would be sister chromatids. And the center here, you have what's called the centromere, the linking portion between all 4 branches in this case. Now, I've shown you this picture before, but again, I like it because it shows the different scale as you coil DNA up. It starts at only about 2 nanometers, and then it gets bigger and bigger as you're coiling around these histone proteins, these circular proteins that are then coiled around themselves. And again, the size is now up to 30 nanometers, and then you start coiling these things even more, and you're up to 300 nanometers, and again around themselves, and eventually you see an entire chromosome at about 1400 nanometers. Now, you can see the size of a chromosome under a microscope at 1400 nanometers. It's very, very difficult to see just a single strand of DNA stretched all the way out at 2 nanometers. So you can see this, but it's very difficult to see that under a microscope. Here's an electrograph image of a set of chromosomes. I've shown you these guys before. Again, you can see the centromere right here, the center point, and then you have the chromatids here. These would be sister chromatids, and then here would be another set of sister chromatids here. And it's important to remember that, again, the chromosomes only show up inside of the nucleus right here, just before the cell's about to undergo mitosis or meiosis, just before the cell's about to divide, because most of the time you want the DNA loose so that you can make proteins, you can do transcription and translation. When the DNA is in the chromosome form, it's very hard to do transcription and translation because it's too condensed. It's packed away, basically. And the reason they pack it away, like I said, is so it's easier to split up the nucleus, to divide the chromosomes into the 2 new nuclei you're going to get once you do mitosis or meiosis. So again, on the basic structure of the chromosomes, the center here is called the centromere, and there's 2 different types of chromosome structure. You can have a single chromosome, like you have here on the left, or a double chromosome. And if we're going to assume, for this instance, that the blue outlined chromatids are the original DNA, when you see a double chromosome form, that means the DNA has already been replicated. It's already been copied. So it's been copied so that you can get 2 new chromosomes for the 2 new nuclei you're going to form. So we would assume that the red, in this case, is a copy. And again, you have a chromatid for each branch, and then the double chromosomes, these are sister chromatids. And then the blue is the original, whereas the red is the copied strands of DNA. Ideally, the copy structure should be as precise as possible. Occasionally errors do happen though, and we'll talk about those types of mutations, but the original and the copy should essentially be the same, coding for the same proteins through the process of transcription and translation. Now, don't confuse the term sister chromatids, which we talked about before in the last slides, with this term, homologous chromosomes. These occur in most sexually reproducing species, animals especially, sometimes in plants as well. This is where a organism gets 1 chromosome for each trait from both parents. So all the genetic traits get 2 sets of instructions, 1 from the mother, 1 from the father. So both parents contribute to the genetic makeup of the offspring. So for example, Here I have two homologous chromosomes. Again, they're from different sources. One came from the father's side, the dad's side, one came from the mother's side, but they code for the same basic thing, even if they have different alleles. Maybe they're coding for three genes, H, W, and C, where H has something to do with the influence of height, W has something to do with the influence of metabolism or weight, and C has something to do with hair color. And so maybe mom's chromosome here, the capital C, is dominant for, say, dark hair, where the dad's lowercase c is somehow dominant for lighter hair. Or mom's metabolism gene codes for a faster metabolism, whereas dad's codes for a slower metabolism. Or then maybe, having a gene that somehow influences body growth in terms of height, and then another one that influences body growth in terms of height, but in a different way. Again, these are different alleles, but they are the same gene. So remember what the term alleles means. And that makes these homologous chromosomes. So two sets of instructions for the same exact trait. Both of these go to the offspring, and note that, again, these are not sister chromatids. Sister chromatids come from the same exact cells, whereas homologous chromosomes are the different chromosomes coming from two different parents that code for the same thing. Now, as I said before, typically the DNA, when it gets copied, is copied pretty precisely, but occasionally you can get an error. We call these errors mutations. And that's any change in the nucleotide sequence of DNA. And the ones I'm going to talk about today are called point mutations. This is a type of mutation caused by an error at a single base pair, a single point in the DNA sequence. And most of the times, the names give away what kind of mutation effect this is going to have. So, for instance, if my original DNA strand was AGCTC, a substitution point mutation might be something like this. We now have a mutated strand that's AGGTC. Now, do you see what's happened? We've substituted in the G where the C was. That's a substitution mutation. Second type of point mutation is called an insertion. Again, we're going to start off with the same normal strand of DNA, A, G, C, T, C, and in an insertion point mutation, you can see we've inserted an A, an adenine, where there was none before. It's just not here. So that's called an insertion mutation. And the third type is called the deletion. And again, the name kind of gives away what's going to happen. We start with the same normal DNA strand, A, G, C, T, C, and in a deletion, one base pair gets isolated and accidentally skipped or missing. And so suddenly the T is now gone, where it was once there. And note that all these tend to change the order of the code, of the genetic code, so that when transcription and translation happen, and this DNA strand is read to be coded for the mRNA, which will eventually be coded during the translation process to make a protein, you might not get the exact protein shape you want. And if you get the wrong shape of the protein, remember that the function might break down. This is why point mutations can sometimes be a big issue. So we're going to go on and talk a little bit about mitosis. I've shown you this picture before. Again, this is a cell undergoing mitosis, and we're going to just briefly run through the various stages. Once again, this should be familiar in basic review, but I'm going to add to it now a little bit. So mitosis, how your body grows and how it repairs. So repair is another big function of, make that a better R, another big function of mitosis. So when a cell divides to make two complete daughter cells, basically clones of the original mother cell, that's mitosis. Mitosis occurs in the set pattern of phases, which you guys should now know from your mitosis handbook, and it's all encompassed in what we call the cell cycle. Remember, most of the cell cycle is interphase, the G1, the S, the G2 phase, and then you have mitosis, and then it goes back to interphase, and the cell never goes out of this pattern unless something is wrong with it, unless somehow the cell is sick in some way. So the basic cell cycle goes interphase, then mitosis, then cytokinesis. Interphase, mitosis, cytokinesis. Whereas the stuff in the black, which you guys learned for your mitosis handbook, is strictly speaking only mitosis. Prophase, metaphase, anaphase, telophase. And remember, the phrase I taught you in the last lecture on how to remember this is, I prefer milk and tea cold. interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Again, remembering that interphase and cytokinesis are not part of mitosis. Mitosis is only prophase, metaphase, anaphase, and telophase. And again, you can see this process repeating. It is a cycle. So I prefer milk and tea cold is a great little system for remembering interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Just remember that interphase up here and cytokinesis are not technically part of mitosis. And remember that in mitosis, cells are dividing to make more cells. This is the process of how your body grows from being a one-celled zygote, a combination of sperm and egg, to the full adult human being you almost are now. And this is also how your body tends to repair itself. So when you ever get a cut or a bruise and your body has to, or you break a bone and your body has to replace cells, it's mitosis that does the repairing job, growing new cells, bone cells, skin cells, whatever you need. Keep in mind, not all cells have the propensity to do mitosis. The one type of cell that usually doesn't do a whole lot of mitosis after the age of 21 is your nerve cells, your brain cells. Those stop doing mitosis once you reach adulthood, and they usually don't repair themselves very well. So some cells do mitosis a lot, like your liver cells. They constantly undergo mitosis. Some cells only do it for a certain amount of time, usually during the growth and development phase of a life cycle. So again, in mitosis, one parent cell, that's 2N, remember this is diploid, gets divided up into two daughter cells, both of which are diploid. Remember, in humans, the magic number for chromosomes is 46 or 23 pairs. So the parent cell has 46 here. Each daughter cell is going to have 46 chromosomes and another 46 chromosomes. And again, this picture is a nice picture because it shows you the single chromosomes right here before DNA replication. Then the DNA gets replicated, so they become double chromosomes in that second picture. Then the cell starts to break apart through mitosis. The chromosomes are split, and you get two daughter cells. And again, because the parent was 2N with two chromosomes, both daughter cells are 2N with two chromosomes. And again, this should look familiar from the mitosis handbook you guys did. Just notice that in interphase, again, the DNA is loose. It's uncoiled. It's in chromatin form. This is the easiest way for transcription and translation to occur. We pack it into chromosomes just as mitosis occurs so that the cell can make it a very easy job to separate all the DNA. So it's not pulling these huge, long strings. So they condense it first and then pull them apart. So then you get two new nuclei starting to form in the last image. Now, let's contrast that to meiosis. Meiosis is the process of how your cells make gametes. And gametes is just a simple term for saying the sex cells. That includes the sperm cells or the egg cells. And meiosis is slightly different because you start off with one parent cell that's diploid, 2N. Again, in humans, this would be 46 chromosomes. And you produce not two, but four daughter cells. And we call these daughter cells the gametes. Again, sperm or egg. And all of these are not diploid, they're haploid. So each of these has only 23 chromosomes in humans. So remember, 2N, diploid, you might see it as 2 capital N, same thing. Diploid, 46 chromosomes in humans. In regular cells, that includes all your skin cells, all your heart cells, all your muscle cells, all your bone cells. Most of the cells in your body are diploid. The only case where that's different is in the gametes, the sex cells. Those will be haploid, or only 23 chromosomes. So nearly every cell in your body has 46 chromosomes except for the gametes. And mitosis, which is, again, used for growth and repair, is contrasted to meiosis, which is used for the process of reproduction, producing the gametes, producing the sperm or the egg cells. Now, this is a very brief picture of meiosis. I'm going to run through the stages very quickly and be happy I never made you do a meiosis handbook because you'll see why. Meiosis is actually broken up into two major sets of stages. You have part 1 called meiosis 1, which includes a prophase 1, metaphase 1, anaphase 1, telophase 1, and a cytokinesis stage. That's the process of going from here to here. And then there's a part 2, which includes meiosis 2, which is technically the name for part 2, prophase 2, metaphase 2, anaphase 2, telophase 2, and again a cytokinesis stage, which is how you go from here to here. So to show this in detail, I'm going to put some labels onto this picture, and I do recommend in your notes getting a copy of this picture down. Again, you're starting with a parent cell that's diploid. In this case, the parent cell has how many chromosomes? 2. So 2n diploid in this case means 2 for this particular cell. You have DNA replication, so the chromosomes which were single become double chromosomes. Then you undergo the first stage of meiosis, meiosis number 1. The cell splits, and notice what happens. Each daughter cell now becomes only a container for the double chromosomes individually. In other words, the number of chromosomes got cut in half. And then you undergo meiosis 2. And again, here's where the double chromosomes become separated. And now look what happens at the end. Down here, each cell has only one chromosome. It's now just N, or N. Haploid, the original number. So on the top you had 2, on the bottom you have 1. The top is diploid, the bottom is haploid. And notice again you have 4 gamete cells, 4 either sperm or potentially 4 eggs. One side note though, when this happens with female sex cells, usually only one cell, like say this cell, will become the egg. The other 4 become non-existent eggs called polar bodies. So it's not perfect, but it does produce 4 daughter cells. Here's the stages of meiosis. Again, starting from the top left, this is meiosis 1 from here to here. You can see the double chromosomes become lined up and then they become separated. And you'll notice right here, there's a part I'm not really talking too much about. It's called crossing over. This occasionally happens when chromosomes spend too much time in contact with each other. They'll actually exchange segments of the chromatids. This is a way of increasing genetic variation within offspring and potentially giving new forms of varieties, thus new adaptations, which we'll talk about in evolution. But again, notice the double chromosomes get split up. They become uncoiled by stage E here. That's the end of meiosis 1. And then meiosis 2 takes over, and again, they line up along a metaphase plate, and these double chromosomes now separate to produce 4 new nuclei, each of which has only 2 chromosomes, where originally there was 4 chromosomes. So this is the 2n, and this is the n. So you went from 4 down to 2, 4 to 2. You went from diploid, 2n, sorry, my 2n's not neat, let's try that better, 2n to just n for each of the cells. Again, from 4 to 2. Here's an electrograph image. Sorry, my writing's still on the picture there, but it shows the process of meiosis in action. Again, you can see meiosis 1 on top, the chromosomes becoming condensed and then separating, and then they turn and they separate yet again, producing 1, 2, 3, 4 new daughter cells. So that's the process of meiosis, the way your body produces the gametes, the sex